If I've forgotten to say hi to anyone, that's not on purpose. I'm not discriminating anyone. My screen gets very busy all of a sudden. But I think we have a recording running, and I was saying before we started already that what I'm planning to do today is to go through quite a few things. So I don't want anyone to feel that I'm rushing. So if I go too fast, you let me know. But I also want to be respectful of your time so we don't run over what we have allocated for today. But just for the sake of recording, it's the week 7 evening. I was going to say afternoon, but it is really evening session. And what we've got lined up is not the blood vessels, although it says there. So what we are looking is going to be the lymphatic and immune system. Clearly, when I was preparing the slides yesterday, I was moving on too fast. Just so that no one's confused. So what I want to do is, like always, I have a little bit of a plan, what I'd like to cover today. And what I really want to spend time is talk about the lab and how the lab is different from what we've done before, once more, in a way that you completed it and in a way that you return it to us. So that's one big topic that I want us to spend time on. The other big topic, of course, is going to be the chapter material from this week's material. It's going to be the immune system and lymphatic system. We cover both. It's quite an ask for 1 week. So I would anticipate that it's a little bit same situation as with the endocrine system, that there's a lot to go through, and to really have a fully educated grasp of everything so that you can say that I've totally got this. I understand exactly what we're talking about. We would probably need a couple of years. So what we'll do our best to have a general understanding of what the immune and lymphatic system entail in our perspective for what our needs are. And I don't want you to feel threatened by that or intimidated if there's some parts that kind of remain a little bit vague, that you're still wondering that, okay, I don't fully understand this. I'm happy to answer any questions and I'll try to fill in those gaps, but just don't let that overwhelm you. And I also want to spend a little bit time today talking about the exam and lab practical, which are coming next week. I'm super excited because really this week we have these chapters, and then we have the exam and lab practical, and then it's already spring break. I can't believe where the half of the semester has gone, but I'm ready for a break. I don't know about you guys. At this point, once we get to the spring break, you have done about half of this course. So keep that on the side. Let's finish the first half very, very strong. And then you pretty much are all set for 50% of the course content and almost 50% of the course points that count towards your final grade. So I'll say a couple of words about the exam and lab practical. And that's really the big gist of this session. So I wanted to start by going through real quickly assignments, because that's what I've been student myself for a long time. I know that's what you want to know, so you know how your final grade adds up. So this week we have all these similar elements that you've seen before. We have the Learn Smart, which was really just reviewing the chapter material with that textbook available there, direct links and answering questions about that. We have the quiz, and then we have the practice anatomy, which is really good practice for the practical. And those are the 3 elements that you are totally familiar with. There's nothing new about those. You already know exactly how they work. The new element this week is going to be the immunology virtual lab. So it's not going to be the exact same format as what you have seen so far with the labs, but I would say that it's more simple. And what I mean by that is that you're actually not completing this virtual lab through the McGraw-Hill, through the Connect. Instead, you're completing it fully through Canvas and the links from Canvas. So let's have a little bit of a look of that, just so that everyone knows what we're gonna do and how it works. So if you look on this week's assignments, you will notice that the lab is all about immunology. And when you click on that lab, a Canvas page opens. And on that Canvas page, you have two links. One of them is to go to the BIO Interactive website, which is a website that's available for you, and it walks you through a lab activity where you learn a bunch about immunology. The other link is to download or open up a PDF file, which is basically a worksheet for this lab. And you can either download it and complete it on your computer, or you can print it and complete it by hand. Or if you don't want to print it, you can just take a normal white sheet of paper, or any color sheet of paper for that matter, and fill in your answers, just number them, and then scan that. Or you can do it on Notepad, whatever works for you. What I'm looking for this lab is to get the worksheet back in one form or another. It can be just your answers. It doesn't have to be the actual sheet. So whatever works for you. But if you decide to do what most students seem to prefer to do, you open up this lab worksheet. And what I want to highlight here, I think you'll all be fine, but I'm just for the sake of being consistent, I want to show that it's 6 pages long. That might feel like, oh my goodness, that's a lot of questions. It actually isn't. And if you look at the questions, I'm not looking for an essay of an answer to those questions, just a little short response to each question is all that's needed. And just let me make a note. So for anyone who joined just us, you haven't really missed anything crucial. I just started talking about this week's lab, which is a different format from what we've seen before, but I'll show it also on the Canvas in a second. But just make sure that you work through that worksheet. This is the component that you'll submit back to me, and this is the component that I'll give you the grade for the lab as well based on. Just a simple answer is enough. Of course, I'm happy to read if you want to write me lots, but don't make it harder than it is. And actually, once I open that lab sheet for you on the screen, you'll also see that it's not quite 6 pages. I think the last page has 1 question and so on. So it's not quite as big of a project as it might initially seem. But once you have that lab sheet, the other thing that you want to do, you want to enter to the BIO Interactive website, which looks something like this. And there on the left-hand side, not right at the top, but close there, you'll notice a button that says Launch Interactive. That's what you'll click, and it actually opens up a whole virtual lab that you'll work through. And as you're working through that lab, you'll get answers to that lab report or lab worksheet. And that's once you have completed that, you then go back to the Canvas. And in the Canvas, there's a space where you can upload your completed sheet. And that's what I give the grade based on for this week's lab. So without going any further, I'm just going to jump to our Canvas course. Ooh, I shouldn't be multi. That's not my strongest point. Hold on. This looks better. So you should be seeing now my web browser sheet or web browser view. And as we go to the modules, provided that my computer is willing to work with me, your view will, as a student, look a little bit different. So I'm just pulling up the view that's aligning with what I'm expecting you to see. Provided that Canvas is willing to cooperate with us. It's been running a little slow today. I don't know if any of you have run into issues with that. If that's been the case, remember that I'm always happy to give extensions if that slows you down. But here we are with what you should be able to see as well. And once we get to the week 7, where we're looking at the lymphatic and immune system, and we scroll all the way to the assignments, you'll see that the immunology virtual lab is kind of indented a little bit in just to make it stand out. You click on that, and we end up to this window where I said that you'll find both the link to the actual lab and the worksheet for the lab. And you just click onto the worksheet and it should download it there on your computer. And I'll show it in a second what the worksheet will look like. You also see this link to the biointeractive lab, which takes you to the very same page that we just saw. You click launch the lab, and I'm hoping that you'll be able to see the same window that I have here, which is really the virtual lab. You'll work through that step by step, and you'll get all the answers to the lab questions that I'm asking you to answer to. So let me just open up another window. I want to show you the lab worksheet just quickly. Here we have it. So you should be able to see a PDF file that I've downloaded. That is the virtual lab worksheet. There's a bunch of questions. Like I said earlier on, don't make life more difficult for yourself than it is. So just answer to the basic questions with the detail that you feel comfortable with. I'm not expecting you to go anywhere outside what's covered in the lab. The level of detail from the lab is more than fine. And as you work through it, you'll get all of those answers. And I think that you'll fill in a couple of tables. And the final page, as I said, there really isn't much on that page. So it's really about 5 pages long. So nothing of that should take you too long to work through. So that's the only difference with this week's lab that I wanted to share with you, so you're not gonna be caught off guard with that. So having said that, I think we're ready to have a look of this week's lecture material, unless someone has a question about the assignments or the labs or anything that we've covered in this first part of the session. Just feel free to write on the chat or unmute yourself, shout out. If I don't hear anything, I'll just keep continuing. But I really do want you to know that I am not running away from questions. I'm totally happy to facilitate any questions that might come to your mind. So, like I said, on this week's session, you're actually covering 2 large topics, the lymphatic system and then the immune system. In many courses where I teach in other places, whether it's a college or whether it's a university, we tackle these separately. You can even do a full degree in immunology and spend 3 to 4 years studying just that field. So there's a lot to cover. And I really feel that really to have a good grasp of everything from these 2 topics, it's outside of what we can do realistically on this 1 week. But I feel at the same time that what often happens is that our colleagues in other schools don't spend enough time on talking about lymphatic and immune system, or they might not spend at all any time talking about these 2 systems. And that's a big shame, because they are important systems for us to be at least aware of. So we'll do the best we can within the time that we have, but I don't want you to feel intimidated if it doesn't feel like everything makes sense. we'll stick to the basics and take it from there. So we're going to start by looking at the lymphatic system. And as we saw in our couple of previous chapters, the cardiovascular system was composed of the blood, the heart, and then the vasculature. And what we end up finding is that in the vasculature, once we get to the capillaries, there's always a little amount of fluids leaking out. And how we are going to deal with that is that we catch that fluid that leaks out and we catch it with the lymphatic system. And just bear with me, I'm going to pause the screen for a second. I just have to make one change to make sure that the slides don't move ahead by themselves. So bear with me. Now we should be better. We should not be surprised by the slides moving along on their own. So what we saw is that if you think of the cardiovascular system as the water supply to the heart, to the house, so you get a water heater or a water pump, you could consider that that's comparable to the heart. You got the water, which is comparable to the blood, and then you've got your pipes, which is comparable to the vasculature. Well, we don't use all the water that we get out of the water system. Some of it is gonna be drained by the drainage system and taken away. And that's, if you want to use that metaphor, that is the lymphatic system in this case. And really the clinical relevance of the lymphatic system that I think is most familiar to most is when you go to the doctor and doctor is looking for your lymph nodes, checking them when you go and very early on the appointment. What the doctor is doing there, they're looking for two things. They're looking whether these lymph nodes are swollen. So we're looking, are they swelling? Are they something that you can notice as being more pronounced, more swollen than what they normally should? And also we're looking whether the individual finds that those lymph nodes, when we touch them, are tender to touch. If this happens, this is a great indication that we have infection going on somewhere in the body. We don't know where. Lymphatic system simply allows us to notice and identify that there is an infection somewhere, but we don't know where. But knowing that there is an infection then allows us on that appointment move our focus on figuring out where the infection is and what kind of an infection and how are we going to deal with it. So that's really what I can think of as the best example of clinical relevance of the lymphatic system. There's a couple of other branches and other things where we can go with this thinking, but we'll stick with that example. I think that what we're going to do next, I just wanted to show you kind of an outline of what I want to cover in terms of the lymphatic system, and this is it. There's not much more than what I've highlighted or listed here. We're going to start by talking a little bit about the lymphatic vessels. Then we'll talk a little about lymph, and finally finish off by talking about lymph nodes. And I think that we spend the most of the time really on looking at different vessels. And lymph gets covered quite quickly, and then we'll spend somewhere in between those two of the amount of time looking at the lymph nodes and lymphatic organs. So what you see, hold on. I wasn't meaning to leave the video there. Bear with me. Let's see. I apologize for that. Everywhere else in this presentation, I got the videos removed, but that was the one that remained there. So I don't want to speak over myself or my virtual counterpart to speak over this session. So what we see here is a picture that's very, very similar to what we saw earlier on when we finished the chapter on the vasculature. And when we finished the chapter on vasculature, we noticed that there were two forces that played role in terms of pushing blood out of the blood vessels and pushing blood back into the blood vessels. Well, the force pushing the blood out from the blood vessel was known as hydrostatic pressure. So that's the force that drives the blood out from the vasculature. The counterforce for that is going to be our osmotic pressure. And normally what we end up finding is that these forces are in balance. So we don't end up losing blood and we don't end up gaining other liquids into the cardiovascular system, into the vasculature blood vessels. So that's the normal situation in a single level of, if we look at on a small level of this. But when we look at the entire cardiovascular system and all of the capillaries that we have there, the balances are not exactly equal. They're close, but not exactly equal. So let's have a look at what happens there. So we have our blood flowing within the vasculature here. We have capillary filtration taking place. And you remember that capillary filtration was this exchange of materials from the blood to the tissues and from the tissues back to the blood. And good examples of that was, for example, the gases, oxygen and carbon dioxide, oxygen being given to the tissues and carbon dioxide being picked up by the blood. Same goes for nutrients and energy. And waste products were typically picked up by blood. Well, if we don't have immediately cells next to the capillaries in the at the side of the capillary network, sometimes we have a little bit of a gap between these cells and between the capillary. All of this space that we have in between the cells is known as extracellular matrix. And that simply refers that there is space that's not within the cell, but it's not also within the blood vessel. And this extracellular matrix is filled with liquid. And the liquid that we saw leaving the blood vessels due to the hydrostatic forces, that's going to be the plasma component of the blood. But once this plasma component of the blood reaches this space, this extracellular space, it becomes now interstitial fluid. So interstitial fluid is, in terms of the composition, the exact same as the plasma of the blood. Its name is different only simply because it's not now anymore part of the blood. Well, this interstitial fluid, if we do the math of how much blood or how much plasma we have escaping from the capillaries in a single day all over the body, it ends up being that there's about 20 liters of plasma escaping the blood. And that was all due to the hydrostatic pressure. Well, about 17 liters get taken back, reabsorbed due to the osmotic pressure. So you'll notice these are values for the entire cardiovascular system in a human being. and they are on a daily level. They're close of being same numbers, but they're not quite. And if my math serves me right, it should. We are left with 3 liters of fluid escaping in total of the cardiovascular system in a single day to this space between the cells. And this fluid is what we described as the interstitial fluid. So fluid in between the cells. Well, we can't have that fluid just building up to this space between the cells outside the cardiovascular system, because otherwise, if this just keeps going on and on and on, we are of course left with edema. The individual will swell either locally or globally, and that's not something that we can sustain. So this is where the lymphatic system steps in. So lymphatic system picks up these 3 liters and transports it back to the blood, back to the cardiovascular system. So knowing all this, we are left with 3 terms that we need to know. And these 3 terms really all refer to the same component of the blood. But and they are, in terms of the composition as well, the same, but we use different terms for them. And this, where we use which one of each of these terms, is going to be important for us to know. So we had blood, or if you wish to be more specific, plasma of the blood, and that's the liquid that is circulated within the cardiovascular system, within the vasculature. Once that plasma of the blood leaves the cardiovascular system, leaves the blood vessels, and escapes to this space between the cells and the capillaries, it now becomes interstitial fluid. So interstitial fluid is this fluid in between the cells outside the cardiovascular system. The composition of the interstitial fluid is exactly the same as plasma. Well, this interstitial fluid, which was 3 liters in total per day in body, approximately, that cannot stay around there. It needs to be picked up by the lymphatic system. Well, we no longer talk about interstitial fluid from the moment onwards when this fluid has been picked up by the lymphatic vessels. So students often are wondering, why do we have these different names for the same thing? It is simply dependent on at what part of these systems the liquid, the fluid is at that moment. And I think that this would make a really good exam question to ask, that when does interstitial fluid become lymph? Well, you know that it would become a lymph at the moment when it enters the lymphatic vessels. So when it is within the lymphatic system, we stop talking about interstitial fluid. Now we talk about lymph, even though it is the same stuff. So I think that that's a good introduction to some of the important terms that we need to know in terms of this chapter. What I want to highlight here is that really we're going to be talking first about the lymphatic system, but after that we will shift our view to the immune system, and you'll see that there's a significant amount of overlap between the two. So you can think of the lymphatic system as the structural part of this interaction between these two systems. It is all of the structures and all of the stuff that circulates there, whereas everything of the immune system is more of physiology. It doesn't have its own structures. It borrows those structures of the lymphatic system. So that was just to kind of put it on the map on the back of your mind, that we'll get to that later on. So what I've got here is a very simplified diagram of what we've got in a cardiovascular system. And of course, we are familiar with the heart. We remember that we could represent it by these 4 boxes, which represented the different chambers of the heart. And we have only included here the systemic circuit. So we would also have the pulmonary circuit, which went to the lungs, but for purposes of keeping everything simple, we're gonna skip the pulmonary system, and we direct our attention fully on the systemic circuit of the cardiovascular system. So blood gets pumped out of the heart to the arteries, then arterioles, then capillaries, then venules, and finally to veins. So what you'll notice about the heart and blood and the blood vessels, the system that it makes up is fully a closed loop. Ooh, I didn't mean to do that. It is fully a closed loop, so no liquid really escapes from this system in terms of there being openings. There's no clear opening for this liquid to escape. So the amount of liquid that we just saw escaping actually escapes through the walls of the capillaries. Another thing that we'll notice, of course, is that this system is one-way system, so blood travels to one direction and one direction only. So all of this should be familiar, but I said that the cardiovascular and lymphatic system are a little bit like complementary systems to each other. So let's add the lymphatic system to this diagram. And what we'll notice that this lymphatic system is in certain parts similar, but in other parts very different from what we've seen with the cardiovascular system. So what's common is that it is a one-way system. Now lymph travels just as blood travels in the cardiovascular system to one direction and one direction only. But what's the biggest difference is that the lymphatic system is open from one end. Rather than being a closed loop, as we saw with the cardiovascular system, lymphatic system starts at the level of capillaries and ends right next to the heart to the major veins that bring blood back to the heart. So it is an open system, open from this end where we see the capillaries. So that's the big difference there. And now we can take all this information that we have, and what we're going to do next, we're going to spend the next, I'd say 10 minutes or so, looking at the different kinds of lymphatic vessels that we find within the lymphatic system. So what kind of vessels we have there? And we're going to start a discussion with the capillaries. And if we compare the capillaries to our capillaries of the cardiovascular system, we'll notice that they're very similar. The big difference between lymphatic capillaries and cardiovascular systems capillaries is that the lymphatic ones are even more permeable than what we saw in the cardiovascular system. And this allows these capillaries to take up larger molecules and particles than what was possible with the cardiovascular system. And you might wonder, why does this matter? Well, I'll tell you one reason why it matters, and this is actually going to be one of the important functions of the lymphatic system. So when you eat food and that food gets digested in your digestive system, so broken into smaller components, and then absorbed into your body, so taken away from that digestive system and actually taken to the bloodstream, and it travels wherever the energy and nutrients are needed. What happens with the cardiovascular system is that cardiovascular system is responsible for picking up all these molecules that we have broken from the food, so all the energy and all the nutrients. However, if we have large molecules, such as, I think a good example of that would be dietary fats. So let's say that here we have a large dietary fat molecule. That is going to be too large for the cardiovascular system's capillaries to pick up. So dietary fats they need to be transported to the cardiovascular system, but they're going to be transported in a roundabout way. And this roundabout way is going to be our lymphatic capillaries. So these lymphatic capillaries are able to pick up these larger molecules than what we are used to with the cardiovascular system, and they transport them right next to the heart, where they then get dumped into the blood in one of the major veins. So really, the pickup and transport of dietary fats is one of the key functions of the lymphatic system. And what I'd like to do now, I'm just going to jump to a slide where I have a clear picture or a clear space. I would like to list with you, before we get further, the functions of the lymphatic system. So we're going to start with the third function, which was the dietary fats. There's two other functions, as I'm sure you could work out. Well, the second function makes sense based on all this that we discussed, that some of the fluid leaks out, some of it is picked back up. If this wouldn't happen, we would have imbalance in the amounts of the fluids in the human body. So we can conclude that the second function of the lymphatic system is the transport of fluids. So the fluid balance, maintaining the fluid balance, is going to be the second function of the lymphatic system. And the first function, the function that's probably the most obvious for us, is going to be fighting against diseases. So lymphatic system, in addition to the immune system, play important role in protecting us against various pathogens that we can have trying to invade our body. So those are the three essential functions of the lymphatic system: fighting against diseases, transporting fluid so that the fluid balance is maintained, and transporting the dietary fats. And this dietary fats is where we left on this diagram. So these were our lymphatic capillaries. That was the first type of lymphatic vessel that we covered. Well, there's going to be larger lymphatic vessels than capillaries if we follow the path of the lymph from the capillary networks towards where it then eventually gets dumped into the blood. And they get increasingly larger and larger as we approach towards the heart. And now that we have talked about the capillaries, the next one that we're going to look at some of the important vessels that we need to know about. And I've got here a picture of our thoracic cavity looking inside from the anterior view. So what we're seeing that we have removed the anterior portion of the thoracic cavity. We can still see some of the ribs at the back of the cavity. And we can see our trachea here nicely, so our windpipe. And we can see our oesophagus, so tube that connects our mouth to the stomach. And we can also see two vessels that were familiar from the past. We can see the superior vena cava, which was responsible for draining the blood from the head, neck, and upper limbs. And we can see our inferior vena cava, and inferior vena cava was responsible for draining the blood from our lower limbs and the hip region. So if we look at the very back of this thoracic cavity, we'll see on the green color there a couple of structures that are going to be of an interest for us. And the first structure that I want to highlight is going to be thoracic duct. Thoracic duct runs the entire distance of the thoracic cavity just against at the posterior edge of the thoracic cavity on top of where we would have vertebral column. And at the bottom of the thoracic duct, we've got this enlargement, kind of a bulging part. And this is known as cisterna chyli. So those would be two terms that we know about the thoracic cavity that I wanted to introduce at this point. Well, there's going to be more for us to know. We're going to move on and look at the third kind of vessels in the lymphatic system. And these are going to be various trunks that we end up finding in there. And these trunks are named based on the body region where they are found. This is probably not going to be a huge surprise to you. But let's go through them. The first one that I want to highlight is going to be the lumbar trunk. So at the lower part of the back, where we see these larger lymphatic vessels known as trunks. Then we also have intestinal trunk. So that covers the region of intestines, region of abdomen where we have our intestines present, so draining from there. And this is, for example, the good example of a site where the transport of dietary fats became important. And just to recap, this was important in terms of the dietary fats because these are larger molecules than what your cardiovascular system's capillaries can pick up. The next one that I want to show here is going to be our intercostal trunk. So intercostal means, of course, costal referring to the ribs, and inter part there referring to the area between the ribs. So intercostal trunk runs between, or intercostal trunks run between the ribs of the thoracic cavity. The next one that I have here is a bit of a mouthful, bronchomediastinal trunk. Well, that covers both the bronchi and also the mediastinum. You remember mediastinum was this place between the lungs. So that gets drained by this trunk. Finally, two more. I have our subclavian trunk. So underneath the clavicle, just as you had subclavian artery and subclavian vein, we also have subclavian trunk of the lymphatic system. And the final one that I want to highlight here is going to be the jugular trunk, draining the neck and the throat region. So all of these are examples of trunks of the lymphatic system. Well, we get even to bigger structures from here. The largest structure that we have of the lymphatic vessels is gonna be our ducts to look at. And there's a couple of ducts that I want to highlight from this same diagram. So let's start with the right lymphatic duct. That drains your right arm and right part of the head and thorax. And where right lymphatic duct empties, it's gonna be our right subclavian. So right subclavian vein, where the right lymphatic duct empties to. And then we also have the last one to notice from here, our thoracic duct. And thoracic duct drains the remaining body parts and empties then to the left subclavian vein. So you'll notice that both of these, the largest vessels that we noticed as the smaller vessels join together and transport all of the lymph to these two main ducts, both of them emptied close to the heart to these major veins. So that's what I wanted to say about the lymphatic vessels. And when they are not doing their job correctly in terms of draining the lymph, what you end up is that this lymph, 3 liters in a day in total, would end up building to a particular area of the body where the drainage would be of an issue. So here we see edema caused by the buildup of lymph. So we talk about lymphedema. So that's the buildup and severe buildup of lymph into an area. How you can usually tell it apart from someone being a little bit chalkier or someone having something else going on, that's usually not symmetrical on both sides of the body. Of course, the other signs and symptoms of the edema also apply. What could cause this? Could be if we have some sort of a blockage in the lymphatic vessels that would drain this area, or if we have had to remove these lymphatic vessels from this area. Those would be probably the 2 most common causes for the lymphedema to build up. What is probably positive about lymphedema, that over time there is a chance that the situation improves. And this is when the alternative pathways to drain that area enlarge and start to facilitate draining of these sites. This is not always the case, but this can happen, so there is hope with that. Now, the second part of our chapter on the lymphatic system that I mentioned that we'd have a look is the composition of the lymph. And if we look at what lymph is made of, we'll notice that there's some good old friends of ours present in there from what we've seen before. So we have, of course, a lot of water. We have a lot of white blood cells and proteins in there. In addition, we've got a few other things, salts, glucose, and fats. And what I would normally do in a face-to-face class with my students, I would ask them to look at and look. This is same composition as our plasma at large, but now we have these other things added in there. But there's one thing that's missing from blood in general. What we should never see in lymph. And that is, of course, going to be our red blood cells. We do not want to see red blood cells in lymph. If that happens, that is going to be an issue we do have a concern to deal with. Well, I guess the next question to ask would be that where is lymph then produced? Well, lymph is produced at the bone marrow, and also at the lymph nodes. So those are the two sides of production of the lymph. What I want to do for the remaining part of the chapter on the lymphatic system, I just want to have a really brief look at the lymphoid cells, so the cells of the lymphatic system. And at large, we can classify these in two groups. These immune system cells that are really the proper lymphoid tissues, and then a bunch of supporting cells, supporting structures that assist for these tissues. And the two proper lymphoid tissues, if you wish, are going to be our T cells and B cells. And you'll hear a bunch more about T cells and B cells on our chapter about immune system. But what I want you to know at this point is that T cells go and mature at the thymus and B cells at the bone marrow. And then we have, at this point, as long as you know that we have a bunch of these supporting cells that help the lymphoid tissues to do their job and survive. So really the last thing for us to look at is that where do we find these lymphoid tissues? Well, lymphoid tissues are found in certain parts of the body where it's going to be especially important for us to filter the lymph and make sure that we are housing these T and B cells. So we can really classify these that the general tissue of the lymphoid system, lymphatic system, lymphoid tissue is going to be everywhere in the body. But then the lymphoid follicles, so nodules, specialized tissue of the lymphatic system, is going to be our lymph nodes and various lymphatic organs. So we're going to have a really quick look of the lymph nodes. So lymph nodes are these kind of a bean-shaped, if you wish, smallish structures that we have. There's hundreds of them in the body. And typically when you have lymph nodes in one area, you'll end up finding a bunch of them in a chain on that area. If you wish to be creative, you can think of them a little bit looking like a pearl necklace on that area. Their function is really to fight any invading pathogens that might try to get into the body. And this can be really broken up into two different parts. How they do that? Well, first of all, they filter the lymph that travels through them. And in addition to that, they screen that lymph for anything that should not be there. And if there's something that should not be there, our immune system kicks in. And immune system is the second portion that we were going to talk on this chapter. So let's have a look of the lymph nodes and where we might find lymph nodes in a bigger bundles at a particular area. And let's start with the cervical region where we find them. So this is going to be the neck area. This is the area that I think that the doctor would most likely try out. It's easily accessible. It's easily palpable. We can feel the lymph nodes there. And if they are swollen, it's really easy to notice. Also, the patient will tell you if they're sore once you feel them. The second area where we find lymph nodes in a large bundle is going to be our axillary lymph nodes. This is, of course, within the armpit. So there's a lot of lymph nodes there as well. We also have lymph nodes at the supratrochlear region. So this is the area just above the trochlear notch. So opposite side of where your elbow is. So we end up finding lymph nodes there as well. We have lymph nodes in an inguinal area, so within the groin area. And for obvious reasons, that might not be the first area that we check in a typical consultation in a medical office, but it is a possibility. We also have lymph nodes within the abdomen, so at the abdominal area, and our thoracic area as well, so at the thoracic cavity. So those are the regions where we would find lymph nodes commonly. And if we look at the lymph nodes, like I said, they're kind of a small bean-shaped, about 1 inch in size, can vary a little bit on the size. What I want to highlight here in terms of the lymph nodes is, first of all, they're enclosed within a capsule, and there's 2 regions of the lymph nodes that we'll end up noticing. The cortex, which has stained nicely on this actual microscopic image that we have stained on this kind of a reddish orangey color. And then the medulla, which is the deeper part of the lymph node, kind of on a green color on this picture. Within the medulla, we have various different sinuses, and sinus is a general term that we use in anatomy and physiology to describe any empty cavity or a sac within an organ or tissue. So within lymph nodes, we end up finding these cavities, these sinuses as well. If we look at the lymph node as a whole, we'll notice that there's a bunch of vessels that bring the lymph into the lymph node. So these are known as our afferent lymphatic vessels. So again, these are terms that are commonly used. I encourage you to know those. When I think of afferent vessels, they are arriving, so A from that, to the structure. Then the lymph travels through the lymph node and eventually ends up leaving through efferent lymphatic vessels. I think of efferent as exit. So I would want you to be aware of those, and you'll notice that the efferent ones are on that concaved part of this bean-shaped shape, whereas the afferent ones are on this kind of a larger curvature, and they're a little bit more numerous in numbers. Well, what we end up with is these swollen, inflamed lymph nodes. So if you think why they would swell, and you think of the function of the immune system, which was the white blood cells that screened this lymph, and if there was something going on, they're going to become much more numerous in amount as well. Well, when we add a lot of material within a tissue, that tissue tends to swell. When a tissue swells above what it normally should be, it does become sore as well. And we do want to find that these lymph nodes, when they're swollen, we would prefer that they're also sore as well. If you don't get a sensation of soreness in a swollen lymph nodes, that might be an indication that there's something more severe going on than just an inflammation. So what I'd like to do next, I would like to go through the major lymphatic organs, and we're going to go through this rather quickly. Some of these you have already seen. So we're going to start with the spleen. And spleen is about a fist-sized structure on the left side of your abdominal cavity, just below the stomach. It's in fact actually the largest lymphatic organ that we have in the body. One thing to remember if you think of the spleen and try to relate it to what you just learned about the vasculatures of the cardiovascular system, or blood vessels if you wish, we had our splenic artery running to the spleen. So that might help you to kind of think about. And if we think of the function of the spleen, we can notice that that's really the immune surveillance and responding if there's something that shouldn't be there. And it's also cleaning the blood of any old or dead blood cells, platelets, and so on. The other function that spleen has is that that's the site where iron and platelets and many other structures are stored. What's interesting in my mind about the spleen, and especially about the spleen of the early development of an individual, let me just change the color. So I've done a lot of work on the field of embryology, so looking at fetuses and embryos developing life. We end up noticing that the spleens in embryo are actually the site where the red blood cells are produced. So before red blood cells are able to be produced anywhere else, in an embryo, it is actually the spleen that takes this duty or this job of producing the red blood cells for the newborn and developing child. So the next one that I want to have a look of is going to be actually the fact that sometimes we might need to remove a spleen if there's a, for example, it's severely ruptured. So we might need to perform a surgical process which is known as splenectomy. And this can at times repair itself, but we also have an option that liver and bone marrow can take over some of the functions of the spleen if necessary. So that covers everything that I wanted to say about the spleen. The next structure that I want to talk, or a bunch of structures, is going to be our tonsils. And we have many tonsils, and they are named based on their location. So palatine tonsils at the posterior oral cavity, lingual tonsils at the base of the tongue, pharyngeal tonsils would be then at the posterior nasopharynx, and tubal tonsils at the auditory tube. If we think of the tonsils, many of us might have had to deal with swollen and inflamed tonsils. Well, this condition is known as tonsillitis. And cause for this can be either viral or bacterial infection, but some kind of an infection that takes place there. And as a result, you end up with a very sore throat with difficulty of swallowing. Lymph nodes are very tender. It is something that's usually very easy to notice by just simply looking to the oral cavity. And some of us who have had constant tonsillitis, so constant inflammation of tonsils, might end up having to go through tonsillectomy, so removal of tonsils as a surgical operation. This is much more painful if you're older when it gets done. So for the longest of time, I'd say usually it was quite common in 80s and 90s. If you had kids who had repeated cases of tonsillitis, that we just decided, let's chop them off, let's make their life easier, let's remove these tonsils, they won't get inflamed anymore. And that was a kind of a common practice for quite a while. But these days we are moving away from doing that, because what we are noticing is that these tonsils are an important part of the lymphatic system, and they might play a role that we don't fully yet understand. So let's not remove something that we might need and we don't yet understand completely. So I'll leave it up to you what you want to make out of that. The next ones that I want to talk a little bit about are the Peyer's patches. Peyer's patches are these little tonsil-like structures located at the intestinal wall, especially on the small intestines and on the more distal, so further away, the end part of the small intestines. Their functions is again the same, destroying bacteria, invaders that shouldn't be there, and they also are a site where memory lymphocytes are housed. The next one that I want to mention a little bit is going to be our appendix. So appendix is the first part of the large intestines, right after the small intestines ends. And what the appendix serves in terms of the lymphatic system and immune system, it is there again to remove, destroy any bacteria that shouldn't be there, and memory lymphocytes are housed there. Well, many of you might have heard that you can have your appendix inflamed. And if you have inflamed appendix, that can become quite an issue. So what we tend to do is that we remove your appendix in that case. Well, if you can have this appendix that can get inflamed and then you get it removed and you can live your life perfectly fine, why would you have something like that in the body then? That's what I think would be a logical, quite justified question. Well, my answer to that is that we don't know for sure, but the best theory at the moment suggests that we don't really need our appendix. What it is an evolutionary remain from the time when humans were eating much more plant-based diet, and appendix was the site where we housed all of the microorganisms that helped us to digest that plant-based material. Well, since we are not eating as heavily plant-based diet anymore, appendix has been over time shrinking and shrinking and shrinking, and I would predict that over evolution it will eventually disappear. But we're not quite yet there. So when we talk about appendix, it's again one of those structures that we quite don't know what it does, and we quite don't know if it actually has a beneficial role in your immune system. So just like I mentioned with the tonsils, we might end up realizing that maybe we should not remove it as hastily as we have been doing in the past. And that brings us to our last structure to look at, which is going to be the thymus. And thymus you are already familiar with from the past. You remember that it was hard to find because it was larger when you were infant and during your puberty, and then shrinks and gets replaced by the connective tissue and fat tissue. And the important thing with the thymus for our purposes is going to be that that's the site where the T cells mature. So we're going to have a look of the T cells in the next section of this week's material. So this is really all that I wanted to say about the lymphatic system. I know that we have already reached the end of our scheduled time for the session. Understand if you have to go, if you have other places to be. What I'm going to do, I'm still going to carry on of covering the remaining part of this week's material, which is going to be about our immune system. And that's going to be much quicker, but for the sake of completed, I do want to talk about it a little bit. I realize that you might have places to be. If that's the case, feel free to move on. You can find a recording. I think I'm going to be able to upload it immediately after this session, so it will be there. But I'm just quickly going to talk you through what's important about the immune system to know. If there are questions about anything we've covered so far, feel free to shout out. Otherwise, I will just carry on. So we will shift our focus now to the immune system and the role of the immune system, how it relates to what we have just learned about the lymphatic system. So immune system plays a role in protecting us against of the pathogens, against various diseases, various things that shouldn't be in your body. So what we end up finding as an important term that you should know when we cover this chapter is the term pathogen. And pathogen is can be many different things. It is something that shouldn't be in your body. It's an invader. It's something that causes a disease. And there's many different kinds of possible pathogens. I've listed some there at the bottom that we can see many different types of pathogens. For example, viruses, bacteria, parasites, fungus, and so on. And again, give me a second as I will make sure that the presentation doesn't jump ahead without us being ready to move to the next slide. This is something that I think has remained by a mistake there. So we should be now back, and you should be seeing again my presentation. So I was saying that pathogens can be many different types. What pathogens do is that they find their way into the body, and especially to a certain tissue or location, and once they've reached their way there, they will multiply in numbers. And this will then result in an infection of that site. Well, we want to know that there's two ways how your body can protect you against these pathogens. So we're going to look at the two branches of the immune system, and the two branches that we'll see are going to be our innate immunity and our adaptive immunity. Sometimes this adaptive immunity is also referred as acquired. And I think that acquired makes more sense to me. I think it's more commonly used in UK and Europe than here in America. So if that makes sense to you, you can use that as well. The big difference between the two is that the innate immunity is something that's very general in nature. It is not specific, and it protects us against many different kinds of pathogens. Whereas adaptive immunity is what we normally think of when we think of immunity. So it's very specific, and it targets specific pathogens that can enter into your body. And that all works through the various lymphocytes, so white blood cells being involved there. So we're going to start with the innate immunity and have a quick look through that. And innate immunity really is the non-specific branch. And how innate immunity works is that we have many different lines of defense, how we can protect ourselves and our bodies against these pathogens that shouldn't get in. Well, the first line of defense against these pathogens it makes sense that would be simply having some kind of a physical barrier. And I think that the best example of that we can think of is, of course, our skin. So your skin forms a physical barrier that stops most of the pathogens from entering into your body. If you want to make an analog to a medieval castle, for example, skin would serve a little bit of a role of these walls around the castle that prevent, if trouble cannot get in the first place, we don't have to deal with it at all. So that's the role of the skin as the first line of defense, as a physical barrier preventing the pathogens from entering. If you have a cut in the skin, as in case of this, now all of a sudden we have a direct route of entry for pathogens to enter into your body. So this is why we really want to treat even those smallest of cuts, because if they are left uncleaned, and if something that shouldn't get into your body is able to reach in through those, that might turn into a significantly bigger trouble later on. Another type of physical barrier inside our body normally. So in the nasal cavity, oral cavity, continuing all the way to the digestive system, also coming up from reproductive tract, urinary tract, we have various mucous membranes there that serve as the physical barriers. The second line of defense that we'll end up seeing is going to be rather chemicals. And based on various chemicals that kill these pathogens or prevent them from entering into our body. And good examples of that would be, for example, sweat, stomach acids, tears. We even tend to kind of wash the urethra, our urinary system's last part before reaching the outside world, so that pathogens that might be potentially climbing up cannot make their way into the body. So the chemical line of defense, or chemicals form the second line of defense against pathogens in our innate immunity. And of course, we can then have various different kinds of cell responses, and this is where the cells are reacting to protect us against various pathogens. And what we're going to do, we're going to go through these different kinds of cell reactions next. So there's, like I said, there's many different kinds, and these rely on recognizing a pathogen, something that should not be in our body, something that isn't right, something that isn't part of our own body, and then acting on it. So the first example, or first one that I've listed of the cell responses here, is gonna be our phagocytes. So phagocytes are white blood cells that basically eat the foreign invaders that might enter into our body. And let's have a look of this process step by step. So first of all, these phagocytes recognize the pathogen that should not be there. Some pathogens will try to hide from these phagocytes, but most often they are not successful. Well, next thing that happens is that our phagocyte binds to the pathogen that it has encountered and takes it inside itself. And there inside itself, our cells have, for example, lysosomes. So you remember these were the sacs of enzymes or acids or various other things that can kill particles. Well, we bring that now to the site and to deal with this pathogen that fuses to the sac where the pathogen is in and kills it. So that's not good news for this pathogen. Now the pathogen has been killed. And once it has been killed, we can actually eliminate the deactivated parts of the pathogen from the phagocyte so it can continue its journey. What we see in the picture on the circle is actually a microscopic image that has been colored on a transmission electron microscope, and it has been colored. It shows the phagocyte eating or pulling these pathogens towards itself. The next one that I want to talk about as cell type examples are going to be our NK cells. NK stands for natural killer cells. And these are white blood cells that kill certain cells and promote inflammation also on the side. So you can consider these almost as kind of the police or cops that guard around, and then if they are needed to, they will act on an invader, on a pathogen. And how they act is that they induce a mechanism known as apoptosis. And apoptosis should be a term that you've heard before. That refers to the process of programmed cell death. So cell all of a sudden goes into this process where it is programmed to commit a suicide. And this circle here shows a picture of a natural killer cell in a yellow color, acting on what I think is here an embryo, if I remember correctly, of some kind. So it is dealing with that as an invader in the body. The next one that I want to talk about, the cellular responses, is going to be the process of inflammation. So inflammation is a response to some kind of an injury to a body tissue. And this injury can be many different kinds. It can be a physical trauma, it can be heat, it can be chemical, it can be infection, or it can be something else. But there is some sort of an injury happening. So is inflammation good or bad? Well, that depends who you ask from. But if we think of the beneficial uses or beneficial purposes of inflammation, we can think of it as promoting healing. And how it promotes healing, I've listed a bunch of things that take place with inflammation here. So first of all, the inflammation of a site causes this pathogen to stop from spreading from that side. So we kind of separate this pathogen to a particular site that let's not let it spread anywhere else in the body. In addition, it can help with cell parts and also removing pathogens, cell parts to be removed, and also removing the pathogens from that side and from the body as a whole. In addition, inflammation helps by alerting our immunity that there's something going on. Now, immune system should get activated. Let's deal with this. And also helps with generating ideal conditions for the repair processes to take place. Well, the next question that I have is that if we look at these two pictures where we have two different kinds of inflammations taking place, what can we see there? We should probably list the signs and symptoms of inflammation. And there's 4 or 5 of these depending on who you ask from, as if we are looking of acute inflammation. So what do we see here? Let's go through that. First of all, we see that the site that is inflamed is red in color. I think that this makes sense if you think that now, as a result of inflammation, we're bringing in a lot of blood to that site. Well, as the amount of blood is higher than normal, it will also show up on that site as redness of the region. The second sign that I've listed here is going to be kind of a warmness of that site. So that site that's inflamed becomes warm. This is again, if you think of this blood being directed to that particular site from the core of our body, so that blood is going to carry heat with itself also to that area. The third one that I've listed here is that we often see these sites being swollen. Of course, go for it and watch the rest of the video. I won't be going on too much longer, and it's going to be uploaded there by tomorrow morning. Have a good evening. So there's the swelling of that site. And if you wonder why it would swell, it makes sense if you think of that we are all of a sudden bringing in a lot of blood to an area where there normally shouldn't be as much blood. Now these cells are pushed apart. That makes any tissue tender and sore. And the next one that we end up seeing is, like I mentioned, that in addition to being swollen, we end up with sore, painful side of the body. There's so much stuff, the cells are being pulled apart. That's not great. And the final sign and symptom that I've listed here is going to be the impaired function. The location, we don't use it as much as we normally would. And I think that the pain and impaired function do make sense if you think of why would we have these symptoms? Well, that prevents us from using a site that is not on its optimal condition. Let's let it rest, let's let it heal, and let's for sure make sure that nothing spreads from that area by letting it recover. So all of these signs and symptoms, if you think about them, are local and flu-like symptoms that we would find in this area. And I think, again, if I think of the exam, that signs and symptoms of inflammation would make a very nice question in the exam. So what we are left with is to next talk about these antimicrobial proteins. So let's have a look of those. Antimicrobial proteins enhance the immune defense by attacking microorganisms and slowing down these microorganisms from reproducing something that shouldn't be there. And the most common ones are interferons, often abbreviated as IFN. And these warn the healthy neighboring cells and activate the system cells, immune system cells, to deal with that pathogen. So their function is kind of twofold. They give us a warning system for anything else around them, but also bring the immune system to an activated state. And the final one that we have to look at here is going to be, of course, fever. What does it mean if we have a cell response of a fever? Well, what is fever, first of all? Well, it is when we have abnormally high temperature in terms of the body temperature. So is it good to have a fever, or is it bad to have a fever? Well, let's think of that through a couple of points that I've raised or I wanted to highlight. Well, first of all, having fever actually inhibits this microbial growth of a potential invader in the body, potential pathogen. In addition, that increases the metabolic events of that site. So metabolism was all the chemical events, all the chemical reactions taking place in the body. So fever increases the rate of those as well. And finally, it also increases the phagocytosis. So this cell-eating pathogen inside itself and killing it and breaking it into pieces. And in some countries, including where I'm from, we rely on fever quite a bit. So we actually generate an artificial fever by going to a sauna. So the idea behind that is that we help with the healing recovery by having all of these benefits of fever by generating this artificial fever. So one of the discussions that's going on in the medical field is that we have for the longest of time now with modern medicine thought that, okay, if you have a fever, we might want to suppress that and get you back to work as quickly as possible. But when we look at all these benefits of fever, one can ask that should we let the person who's got fever have that fever, stay at home, get better, and then go back to work? Of course, if the fever gets too high, usually I would say too high is close to 40 Celsius degrees. In that temperature, your enzymes start to get breaking apart, so that would not be good. But if fever is less than that, but it is still a clear fever, maybe we should write it out. That person should take the time off and get better with body's natural healing processes. I'll leave it that up to you, just putting it out there, that that's something that's being discussed. And the last thing that we need to look at in terms of the immunity is this second branch of the immune system, which was all about adaptive immunity, or like I said, we call it acquired immunity in Europe. And I think the word acquired makes a little bit more sense because that really explains what's happening here. You get infected once, you build an immune memory about that. That's what we think of as an immunity. So if you meet that pathogen again, you already have a plan in place of how to deal with this pathogen. So we remember that adaptive immunity was very specific. So it is something that we must have for only a particular kind of pathogen. So these are made of highly specialized cells and processes, and what we're going to spend our time on is going to be the B and T cells that we're going to look at. So if we talk about, we looked earlier the innate immunity, and now our focus is moved to this adaptive immunity. We notice that not only it is more specific, it is also slower in its initial response. And this is where the B cells and T cells become important. So these then further specialize to a specific type of cells. And we'll also notice that there are some cells that are shared both by the innate and our adaptive immunity. So let's carry on and have a little bit of a look of the adaptive branch. So as we noticed earlier on, a very specific way of dealing with a pathogen or abnormal cell that shouldn't be there. So what happens here is that we amplify the inflammatory response, so this inflammation that we just discussed about, and we activate processes to deal with it. The problem with the adaptive immunity is that you need to have met the pathogen at least once before. So it works really well only on the second time when meeting this pathogen. So we talk about the adaptive immunity having a need to be primed. So needing to meet this pathogen before, develop a plan to deal with it so that it can work efficiently on that. So the benefits of the adaptive immunity is that it is very specific, and in addition, it doesn't rely on acting on a particular location. Instead, adaptive immunity acts throughout your entire body, so it is systemic in nature. And probably the most useful characteristic of the adaptive immunity is the fact that we develop an immunological memory against that pathogen. So if we meet it again, we're able to deal with it because we have a pre-made plan against it. And there's two branches, two parts to this adaptive immunity. Humoral immunity refers to the fact that we have specific antibodies that we use. So we use these antibodies to recognize foreign substances that shouldn't be in the body. And these circulate free in the body fluids, and when they find an invader, they bind to it. The cell-mediated immunity instead relies on lymphocytes, which are the white blood cells in the lymphatic system, and them acting. These can act directly by killing the infected cell or calling other lymphocytes to deal with those. So they kind of, if you think of it this way, they tag a pathogen in your body that, hey, this needs to be killed. So there's a couple of terms that I want to be sure that we feel comfortable here. And let's look through those. So the first of all, I want to talk about a term of antigen. What is an antigen? So antigen is something that shouldn't be in your body. It's a foreign substance that invades your body. So it becomes the target of this immune system and immune response. So it is not normally present in the host's body. So that's the bad guy, the pathogen that we have now in the body. That's our antigen. Antibodies are triggered as a result of a particular antigen being present. So antibodies are responsible for recognizing and acting on these pathogens. And most commonly, these are known as immunoglobulins, abbreviated as Ig. So we're testing of those in terms of do we have antibodies against a certain pathogen. So these, if you wish to look at them visually, could be illustrated like this. The antigen, you remember, that was the pathogen coming into our body that shouldn't be there. And antibodies were these particles that are responsible for recognizing the antigen and acting. And these two need to be a good fit. Only specific antibodies will bind to specific antigens. So there needs to be almost like a key and lock fit between these two. Antigen binding site must match with the antibody. And your textbook shows the antibodies always as kind of these Y-shaped structures that you get told about. And this is very common, but I do want to mention, this is not something you need to worry about, but I do want to mention that there are many, many, many other kinds of antibodies. Your textbook cuts a couple of corners straight. Here's some of the examples of other looking antibodies that we have, and there's a bunch more. But I just want to put it out there that in reality, there's a much more variety than what you've seen so far. And now I just want to mention really quickly to kind of wrap up the different cell types of the adaptive immune system. So we have our lymphocytes. So these are going to be our B cells and our T cells. And in addition to that, we have something else known as antigen presenting cells that we'll talk about in a bit. They play more of kind of an assistant role in this entire process. But I want us to focus on the B and T cells now to start with. So what happens with B and T cells life cycle is that initially both arise from the red bone marrow, and then B cells go to bone marrow and T cells go to thymus to mature. And during this maturation, they get tested for their functional accuracy. So we make sure that they don't attack your body, and we make sure that they work how they should be. So there's a selection process taking place. And after that, these B and T cells travel to their side of the secondary lymphoid organ. And these can be, for example, the lymph nodes, spleens, tonsil, appendix, thymus, or mouth. And you've seen about those a little bit earlier on. Well, after that, once they have matured in there and had a chance to finish their development, they start circulating in the body. They will meet an antigen and causing B or T cell to continue its development at this point. Well, now these cells multiply in numbers and become activated. So they can act on the invader directly, and also some become memory cells. So in case the same antigen is met later on in our lifetime, we are able to act on it quickly. So that's the process of a life cycle of B and T cells. And we are left with talking about the antigen presenting cells to kind of wrap this up. What antigen presenting cells do is exactly what the name suggests. They meet an antigen but cannot act on it themselves. What they do instead is that they alert the rest of the immune system by communicating that, hey, there is an antigen here. So you can consider them a little bit like helpers of the immune system. So I think that there's different kinds that we can see of antigen presenting cells. There's the dendritic macrophages and B cells, but this level of detail is all you need to know. And if we kind of want to recap, I have uploaded this slide where we compare the cell-mediated and humoral immunity, if that's something of an interest for you. The very last thing that I really want to talk with you guys is going to be in terms of the primary immune response and secondary immune response. So primary immune response refers to when we first encounter a particular pathogen. So this is the first exposure to it. And normally this takes place somewhere between 3 to 6 days after you have met this pathogen. So this is the time when you start feeling sick, approximately from 3 to 6 days after this encounter. And you probably reach the peak of this response around day 10. So we're talking about days. And the amount of immunoglobulins that we have present is rather low. Let's compare that to our secondary immune responses. Secondary immune responses is that when we have met this pathogen earlier, and now we meet it again at a later time. So we have this memory against of a particular pathogen. So lag time before you start feeling sick, before we start acting after this encounter, is rather hours than days. And the peak response takes place somewhere around 2 to 3 days. So it's much quicker. In addition, the response that we get is much stronger, and it's also long-lasting response. This response can last for weeks or even months. And this is all thanks to these memory cells that we have developed for this particular pathogen. I think that explaining it verbally like that might be a little vague. If you're kind of like me, you prefer visual presentation. So I did that exact here. So the primary immune response that we talked about, which took between 3 to 6 days to start when you start feeling sick, and the peak time was around day 10, is shown here. What you'll notice that the amount of antibodies that are produced, I've just kind of made an imaginary exponential scale, since we are not really specifying what this pathogen would be. It is a rather small amount of antibodies that we produce. Well, let's add to this diagram a secondary immune response. Now we have already seen once this pathogen, we've dealt with it, and we have developed these memory cells, if you wish. And now the second immune response, we're talking rather of hours than days for this response to take place. And the peak takes place really soon. So roughly 2 to 3 days after the encounter. You'll also notice that the antibody concentration, so the amount of response, is much higher than what we saw before. So first encounter is weak, takes a long time for that to happen, whereas the second encounter is really strong and really fast. And how we can achieve these can be either naturally, where you are exposed to a particular virus or bacteria in your normal life, or we can receive these artificially. These would be, for example, the vaccines. That's one of the big topics that we talk about now. The other way we could get those would be through mother's antibodies, for example, through the umbilical cord or through the milk from the mother to a newborn infant. Of course, we can then have passive immunity, where you would then could, for example, inject serum for artificially acquired passive immunity. Well, how is this all relevant to us? And this is where we're really going to kind of start wrapping up this session, of course. So organ transplants is the thing that I can think of as the biggest example of when the immunity matters. So this is when we have an organ that's removed from one body and placed into a recipient's body. Of course, we can do some transplants from the same body. This would be, for example, with blood transfusions or skin transfusions from self to yourself. Like I said, the most common one is allografts, where we take the transplant from different individuals of same species, and sometimes we can even do that from one species to another. For example, we use a lot of pig heart valves for humans. We also use certain parts from the monkeys. But why is this an important theme for us to consider? Well, let's get to that in a second. Just first, I wanted to show you how much organ transplants take place. This is a little old data, but I think that the ratio of the cases is roughly the same. So kidneys are probably the most common organ transplants that we do. And the reason why they are so common is because you can live with just one kidney, so it is easy to get donors, and really that's not too difficult of a surgical operation. The second most common organ transplants in the US on the year 2000 were liver transplants, and I think that this is still the case roughly. So liver has amazing regeneration ability. So you can chop off part of a liver and it will return back, it will grow back and become functional very, very quickly. So donating liver is kind of a safe and easy thing to do. Well, next two on the list, the third and fourth most common transplants that we do are hearts and lungs. And you might think that, well, they're not easy. You don't get another heart, you don't get another set of lungs. That's true, but they're so common because those are so vital. You can't live without a heart, you can't live without lung. So we do those transplants because there's no way to get around with not having functional ones of those. Well, I would ask then that, okay, if these are common, how successful are they? And this is something that you have to ask yourself, what is success? How do you define success? So what we'll see that initially most patients survive really well. The transplants survive within the patient really well. But in long term, they are less and less successful. In 10 years, about 50% of patients end up rejecting the organ that they have received as a transplant. So how does this all relate to what we were discussing? Of course, that relates in terms of the organ transplant rejection. So this is when you end up with rejecting something that we transfer. So this can be due to poor fit with the similarity of tissue types, blood types, antigen types, many different issues there. So what we end up having to do to have successful organ transplants is that most of the time we need to knock down or knock out the recipient's immune system so that the immune system of this patient who receives the transplant doesn't start attacking that transplant that they're receiving. So these patients who have received an organ transplant are going to be very vulnerable for many normal pathogens that we normally face in our day-to-day life, and simple infection can be lethal to them. So that's where the immune system and knowledge of the immune system becomes so important. I have just a few more slides left. So first of all, I want to talk really quickly about immunodeficiencies. This is when you have impaired function of the immune system. Your function of the immune cells is not what it should be. An example of that, what I've listed there, the two ones are Hodgkin's disease, which is the cancer of B cells that then results in depressed lymph nodes, and of course AIDS, HIV. In AIDS, helper T cell activity is affected, and this then is inhibited or is shown through various signs and symptoms, weight loss, night sweats, lymph node swelling. And what happens most of the time with AIDS, HIV is that it is the infections that end up killing these patients, not the HIV or AIDS themselves. I've spent a lot of time on this field through my job, and even though we still don't have a treatment for HIV/AIDS, We do have good antivirals that we can use. Many patients are able to live with undetectable viral load, and we have some promising anti-exposure medication already being used, and hopefully one day we are able to deal with this with something that's even better solution, such as vaccination. Well, I also wanted to say a few words about the autoimmune diseases. So autoimmune diseases is when your immune system attacks itself. And examples that I have for that are going to be the rheumatoid arthritis. So, of course, that destroys the joints. Myasthenia gravis, where the nerve muscle connections are affected. The MS. In MS, the myelin, the insulation layer of the nervous system or nerve cell, is affected. In Graves' disease, we see hyperthyroidism, type 1 diabetes mellitus, so the pancreatic cells are destroyed. In lupus, there's a bunch of organs that are affected, and of course then various kidney conditions as well. So how do you treat autoimmune diseases? Well, what we end up having to do is similar that we saw with the organ transplants. We need to somehow suppress the immune system. So we need to somehow knock it out. In addition, we can use anti-inflammatory drugs, and there's much promising research going on this field. So I hope that this situation will be much better in the future. And the very last concept that I want to mention is going to be various different kinds of hypersensitivities that we can have. So this is when your immune system responds to something that is a harmless thing in your body with out of proportion response. So there's an over-the-top response to something totally normal. And two examples that I have here are allergies and anaphylaxis. So anaphylaxis is something that we often need to have to treat with EpiPen through the epinephrine and so on. So that's all that I can think of that I could give you in this amount of time. And I know we ran over time, but as a kind of a walkthrough of what's important of lymphatic and immune system to know. Like I said, I wouldn't blame you if you felt that was a lot and you don't have full understanding of how to piece it all together. This is a review. This is the basic concepts. So don't beat yourself too hard that you should know everything about everything. That's not possible in this amount of time. But you should have a general appreciation that both lymphatic and immune system are important. And what I'm going to do now, I'm going to jump back to our weekly presentation. And we already talked about the lab, and we've covered the key points of this week's material. All that is left for me to talk with you is that next week we will have our exam 2 and practical 2. They are available for the entire week, so you can pick the time when you want to complete them. Remember that during the exam week, I always offer 4 review sessions. On Monday, 1 in the morning at 10 o'clock and 1 in the evening at 8 o'clock. And on Wednesday, 1 in the morning at 10 o'clock and 1 in the evening at 8 o'clock. During these review sessions, I go through the study guide with you. And on that study guide, there are gaps. And this is the only time when I will provide the answers to this study guide. There are going to be recordings available as well, but I don't post completed study guide. This is because all of the questions on the exam are from that study guide, and the study guide covers only the material from the lecture videos of these chapters that we are covering in the exam 2. So that covers the chapter on heart, blood vessels, and today's chapters, lymphatic and immune system. There's going to be about 50 multiple choice matching true false ordering questions, and you have about an hour to complete those, unless you have an accommodation plan in place. In that case, you will have whatever time has been allowed by the student disability resources and services. practice. Remember that the exam is going to be open book. You can take it from home, so you can use all of your materials. I would definitely have the study guide handy and around. I would make sure I would have my notes and textbook available as well. Remember, I never ask anything that wouldn't have been covered in the lecture videos. I don't think there's going to be an extra credit question in the exam, but if I, for some reason, would happen to add it there, I would let you know. But at this point, I don't think there's going to be an extra credit question. The second part of the exam week is going to be our practical. And the second practical, again, is available for you for an entire week. And I really encourage you to make sure that you complete those practice anatomy elements of homework. And before taking the practical exam, go through that practice version of the practical exam. And you might want to go through it a couple of times so you see all possible questions that might come up. Again, the format of the practical is as you've seen before. There's going to be 50 questions that are filling the blank. Don't worry about the spelling. Initially, your score will be something. Once I've graded it manually, it will be different. So I never deduct points about spelling. I also give partial points for good extra information. So that might be something that is worth doing if you don't know the real answer. Again, for 50 questions, you have 60 minutes unless you have an accommodation plan. And in that case, you have whatever has been specified on that. Remember, also the practical is going to be open book test. You can take it from home. So I really encourage you to make use of those practice anatomy and especially of the practice version of the practical 2 on next week's module. Make good notes, take screenshots, do whatever works for you. I know there's many approaches students have used. But the practical is all about identifying structures. There's going to be histology. There's going to be major lymphatic vessels and blood vessels to also identify. And the couple of last things that I kind of say every single time just to remind you is that I am happy to open up any assignments that you might have missed and really want to complete, or if you didn't do as well as you wanted, or if you need extra time. All you have to do is to message me and remember to include specific details of what assignment you're talking about so I can get that taken care of you quicker. And finally, I also wanted to share with you, as before in previous weeks, that you do have online tutoring available through the Cranium Cafe. You can pick the tutor that works with your way of learning, and they will work with you on that online format. The second option is to contact your instructor. So recently, I've been having quite a few sessions with the students from this class. So I'm happy to take the time aside, go through topics or questions that you have, or areas that you feel that you want to review again. A study guide if you have any questions, any technical issues, anything that you're unclear of. I'm usually available to make the time real fast. All you have to do is reach out to me, and usually that can be organized. If not for the same day, for the next day. So just let me know. Usually the quickest way is to reach out via Canvas messages, but I do check also the emails. So that's really all that I had for today. I know it's been a super long session. I appreciate you both sticking around, and trust me that I will not forget that when it comes time to look at where we end up. And same goes for anyone else who joined me today. I do always, when I assign the final grades, look who's been attending, and that counts as a very, very positive thing. So for example, if you're in between of the 2 grades, if you've been attending to the sessions, you can count on me being like, yeah, you should get that higher grade. Of course, there's no requirement to attend. I post these videos as well, so you will not lose points if you don't attend as well. If you have any questions, I'm super happy to answer to those now. If not, really all I have left to say is from this listing that we had a look of before, that do not feel bad if this chapter on the immune system and lymphatic system felt like a lot and you're still kind of like, oh, I have a general idea, but I don't have all the points clear. That is the best we can achieve in this amount of time, and that's enough. I will not test you on great detail on this chapter. So if you have any questions, I'm here. If not, that's all I have for today. I really appreciate you joining me, and I do apologize that today took much longer than I had anticipated.
Hi, Miss Sam. How are you?
I'm doing very well. Is there anything I can help you with?
I just want to say thank you for the support, and also I have one question regarding my grade on the exam, but
And just to finish off this recording, so we were just able to solve a couple of questions that a particular student had. So as always, I encourage you to reach out to me if you have any questions, whether it is about a particular content of a chapter, whether it's that you're stuck with an assignment, or even if it's a technical issue with the connect or any other aspect of this course. I am here to help. That's what I want to do. I want to make sure that you get everything solved, that I can be of an assistance for you, and I want you to do really well on this course. That's my job. That's what I'm here to do. So let me know how I can help you. And thank you again. I know that it's been a super long session today. I hope you have a very good week, and I look forward to seeing you next week on the exam review sessions. Until then, bye.